Congratulations on passing a short MCQ test. Before beginning with the next section on modern-day diagnostic tools, let's have a quick recap of the previous lesson. In 1999, Stuckey in his article stated that unfortunately none of the currently accepted caries diagnostic methodologies have the ability to account for the dynamics of dental caries. He continued to write that including the possibility of reversal, and clinicians are forced to measure a dynamic process using clinical criteria and tools, which are becoming less useful. Not less useful in general, but less useful for these particular problems. This was in 1999, so it was quite a few years ago. In 2015, Schumann in an article stated that a lot has changed since 1999. So, in order to properly diagnose these hidden carious lesions, we need tools that can accomplish this difficult task. And we also need new procedural guidelines for diagnosing and treating these lesions. So let's follow these steps. Number 1, diagnose hidden caries. Then, access the lesion and identify the hidden carious lesion. Then, remove the caries with confirmation, and lastly, restore the tooth. Beginning with the first step, that is diagnosis of carious lesion, we need reliable diagnostic tools to establish the presence of non-cavitated carious lesions. So first, we will discuss light and the various tools which use light to diagnose non-cavitated lesions. So the premise behind using light is that all teeth are translucent. They have specific optical properties. In 1922, Dr. William John Cameron published his work Diagnosis by Transillumination, a treatise on the use of transillumination in diagnosis of infected conditions of the dental process. So that was back in 1922. It's an old concept. Caries detection using a light source to exploit these optical properties is not a new concept. In his pamphlet, he states that this use of transillumination provides one of the most valuable aids in an operative procedure. It can be used effectively for the detection of interproximal caries and intercoronal caries. Caries will at once evidence themselves by a distinct discoloration in the crown of the tooth covering the area affected or filled. He also developed the Cameron dental lamp, showing in the picture in this slide. This slide here shows the various newly discovered light diagnostic tools. It gives an idea of how important correct diagnosis of oral caries is, leading to so many new discoveries. So when we talk about caries detection by laser fluorescence, one of the original devices used was cavodentinogen. In order to use it, the teeth has to be dry and free of plaque, calculus, and stains. So the way this device works is it can read 2 mm into the teeth. The major issue here with this tool is that it is highly sensitive and low in specificity. By this, I mean that it will detect any abnormality and not just caries, but the readings generated are not very specific. So a practitioner, you will have to judge whether reading is coming from tooth decay or any other abnormality. Caries detection using a fiber optic transillumination. Basically, all teeth are translucent. They have specific optical properties. Caries detection using a light source to exploit these optical properties is not a new concept. It can be used effectively for the detection of interproximal caries and intercoronal caries. This is the picture of the Cameron dental lamp, which was invented in 1922. And this is the statement from his book, which says that caries will at once evidence themselves by a distinct discoloration in the crown of the tooth, covering the area affected or filled. Fast forward to 2015, the other picture is a micro transilluminator, very much modern version of the Cameron lamp. So let's have a closer look at fiber optic transillumination. So basically, these devices use a high output of LED to detect interior and posterior caries. So what these devices are also useful for are to detect coronal fractures and root canal orifices. As mentioned in the previous slides, it can detect interproximal caries as well using accessory tips. The correct way is to just put the tip in the center of the teeth and not poke it through and through between the teeth. And the way it works is basically it darkens the area denoting caries, and this is substantiated by the radiographic exam here in this picture. Here in this picture, you could see carious lesion distal to a premolar, and it's substantiated by a fiber optic transilluminator. Here, you could see a class 2 diagnosed by the fiber optic transilluminator, and after the caries has been excavated, you can see depth of the lesion in the second picture. Let's talk about the second device, LED spectroscopy. Spectroscopy is the study of objects based on the spectrum of color they emit, absorb, or reflect. In dentistry, a spectroscopy is a device that emits autofluorescent light and evaluates the fluorescing porphyrin by products of kariogenic bacteria such as Streptococcus mutans, bacteroids, and Actinomyces odontolyticus. So what is a porphyrin? In biology, porphyrin is a class of pigments whose molecules contain a flat ring or four linked heterocyclic groups, sometimes with a central metal atom, and can therefore be detected by sensitive fluorescence measurements in the red spectral region. So that's important. These bacteria give off porphyrin, and porphyrin can be detected by laser fluorescence in the red spectral region. With LED spectroscopy, porphyrin-producing kariogenic bacteria have a unique spectral response to autofluorescence, producing a red color. In contrast, healthy tooth structure will fluorescence green. In a study by Lennon et al., it was determined that red fluorescence is well suited to detection of the bacteria that causes dentine caries. This has been substantiated and repeatedly demonstrated in numerous studies. So there are a whole lot of studies which confirm that LED spectroscopy can detect bacteria which are actually the real cause of carious lesions. But this phenomena occurs in a wide range of bacteria and is not specific to bacteria involved in the pathogenesis of caries only. Perfect example is human skin. If a patient has caries, Propionibacterium acnes sitting on the skin can also be detected by LED spectroscopy. In the same way, high concentration of the porphyrin-producing bacterium Pseudomonas aeruginosa have been found in the biopsies of oral squamous cell carcinoma and exhibit red fluorescence. And based on studies by Koenig et al., it is apparent that the spectral characteristics of bacterial fluorescence can be used in the diagnosis and treatment of a variety of diseases. In fact, many researchers have gone so far as to destroy by exciting the porphyrins contained within them through phototargeting these metabolites with spectrum-specific light. So we're actually at a point where we are not using it only as a diagnostic tool, but also clinically to correct it. So let's have a closer look at the LED spectroscopy. Here I have picked up a specific device, the Spectra system. It uses a 405 nanometer LED and emits an autofluorescent light that is absorbed differently by various objects on and within a tooth. So if you take a look at the top three pictures, the first shows a faulty tooth with an intact enamel but slight discoloration. The Spectra device confirms in the second pic that ocal caries is present. See the red in the Pitt and Fisher areas? Third picture is a cross-section of the same tooth which confirms presence of carious lesion. Lower three pictures is of a sound tooth. No discoloration in the first picture, no red light by LED in the second picture, and no ocal caries in the cross-section in the third picture. To be more specific about the colors, we have a whole color chart which tells us what color means what. Green color denotes sound enamel. Yellow, orange, red is established caries and requires treatment. Blue is the beginning of enamel caries, requiring only sealant application. These two pictures are of the same premolar. First picture was taken by an intraoral digital camera showing slight discoloration of enamel. Next to it is a picture by another type of LED device called Camera X Spectra, which shows red area in Pitt and Fisher, confirming that the discoloration is really by carious lesions.